PhD advisor was Santolini, and what he's going to tell us about today is his PhD work essentially, I understand.
Absolutely. Okay, so thank you for the invitation. So what I'm going to talk about contains a lot of biology. So at any time, please feel free to ask any question if something is unclear. But behind the biology, basically, it's a pretext to apply tools from physics and from machine learning as well. So these tools you may be more familiar with. So maybe, first of all, I'll give you a big picture of what this is about and what the goal of all this work leads to. Here is a complex multicellular organism, okay? And, well, a basic observation is that we have different cell types, okay? These cell types are shown here, okay? Fat cells, heart muscle cells. Though these different cells, they contain the same genome. So from the same genome, they are able to express different set of proteins. And the mechanisms that underlie the differential expression of different sets of genes are regulatory mechanisms. So these regulatory mechanisms, they happen at all scales, at the gene level, so the expression of the gene, at the RNA level, which is the RNA transcribed from the DNA where the gene is, at the protein level. And here I'm going to concentrate on transcriptional network, which is regulation at the level of the expression of a gene. Okay, so to give you an idea of the mechanisms in place, here is a schematic of the genome. So we are in a cell, in the nucleus of the cell. Here is the genome. There are here in green coding regions. So this is a gene, which is coding for a protein, and we know that these coding regions represent some percent of the genome. And in black, you've got non-coding region. And in those non-coding regions, proteins that are called transcription factors can bind DNA on specific sites that are about 10 base pair long. They can as well combine on modules that are called cis regulatory modules or enhancers, and I will often use the term CRMs for cis regulatory modules, that are about 1 kb long and that constitutes functional regulatory sequences with a precise spatio-temporal output in the cell, in the organism. Okay? And these regulatory elements, they can be distant to the gene, they can be upstream or downstream, and they can be very near 1 kb or maybe a megabase away, so very far away. And these regulatory elements interact with the transcriptional unit, which then allow to read the gene, transcribe into RNA, make a protein. Okay, so this is a basal mechanism of regulation, of transcriptional regulation, and these kind of mechanisms underlie the transcriptional network. So basically, what is a transcriptional network? You've got a transcription factor, a protein, that binds DNA, activates another transcription network, I don't know, this one, which in turn activates another transcription network, and so another transcription factor, and so on, thus creating a network. So people have tried building such network, and to do that, they have used things like gene co-expression network, where you look at the correlation between expression of genes to infer some link between transcription factors. But basically, this is just correlation, so you don't really have a direct link. People have tried to use the proximal binding around the genes on very nearby the transcription start sites, but relying on simple models of binding and without the knowledge of the rest of the genome. So models to build this kind of network are, for the moment, simple and quite need to be improved. And the reason why studying such network is important is that it has been recently shown that if you look at the mutations, the single nucleotide polymorphism, so the mutation in your genome that are related to some traits. So GWAS means genome-wide association studies. So the mutations that lead to disease or common traits, about half of them affect not the proteins, but regulatory elements. So they affect the regulation. So one needs to understand in this network where the arrow points in the genome, where are the sites. Okay, and why it's important for diseases, here is just a very simple example. Here you've got a CRM, or an enhancer, so I was presenting before, that regulates a gene 1 megabase away, so quite far away in the genome. You can see the expression that is led by the enhancer here in the limbs of the developing mouse embryo. And when you delete this enhancer in the genome of a mouse, the resulting adult mouse don't have limbs anymore. And in human, there are mutations that happen in these enhancers, and this mutation leads to what is known as polydactyly, which is the presence of supernumerary digits in the hands. So basically here is an example, and one would want to better understand, localize, and find this enhancer in the genome to well, associate with disease and build the whole network. Okay, so biologists have already used some techniques to do that. A technique that I'm going to speak about a lot is called ChIP-seq, for chromatin immunoprecipitation followed by sequencing. The idea is to use an antibody specific of a transcription factor to get the transcription factor along with its bound sequence. So this is a genome that is coming from cell extracts, so from a tissue that you want to study. So this is in vivo data. You purify the DNA that was bound, and you align it to the genome, and you get such data here. So here is a 50 kV region of the genome. You've got the NANOG gene, which is known to be important in stem cell fate. And just upstream of that gene, you can see peaks that correspond to binding events for different transcription factors that were found using ChIP-seq experiments. So here you can find that there is a clustering of different transcription factors, therefore a regulatory element. However, it's not that simple, because in that case, well, it's not because you've got a bound protein that actually there will be a regulation associated to it. It's not straightforward. You do not know which gene is regulated, so you know what is incoming, but you don't know what is the output, where goes the output, where goes the regulation. The bindings that you see can be indirect, so you can be bound because you are bound to another protein that is bound. And the precision you get is around 200 base pair resolution, which is too big compared to the size of the binding site. You don't have all this information, and you want to go further. So people have developed simple models, and this is the most simple models that have been developed. And I will show you how simple it is afterwards. But basically, this is called the position weight matrix model, or PWM model. So this is a second terminology you have to remember, PWM. PWM is this kind of thing. So it's a simple model where you use the knowledge of some binding sites that you know are bound. Okay, so these binding sites, you know they are bound by a transcription factor. You build a frequency matrix corresponding to this binding site, frequency of the nucleotides at each position. And then you take the logarithm to build a matrix that is called the position weight matrix. And for physicists, this corresponds to a matrix of affinities of the protein to the nucleotides at each position in an independent Boltzmann model. And you can represent such a matrix by a logo, where the height of a column is the relative information contained in one column of the matrix compared to the frequencies in the background. So it's basically, here you've got a lot of information because you've always got a C, whereas random generation you would expect 1/4 of a C. And the height of the letters are the frequencies. Okay, so with such a model, you've got an energy matrix that you can use to give a score to any sequence, and thus you can predict if a DNA sequence is bound or not. Yes?
What was feedback in the formula?
Absolutely. So here you compare the observed frequency of a nucleotide in the known sites to their background frequency in the genome. So this is basically, this is the log likelihood ratio between the probability to be bound by the factor and the probability to be generated, randomly generated in the genome. But I will speak more about that just afterwards. And another question, maybe? Okay, and then, okay, you can use such models to scan the genome. You've got predictions for binding sites in the genome. For example, you can take 4 different models. And then when binding sites cluster in one region of the genome, you predict that there is a cis regulatory module, so a regulatory region. You can look in other species if it's conserved. So if it's conserved, that means that there is a function associated to it. And then you can test the sites. But here again, there are problems that you need a good model to predict the sites, and you will have a lot of false positives. You are also limited to what is already known, so the factors for which you have already a model. Okay, so it's not exhaustive. And it's from in vitro studies most of the time, so it's not perfect. So here I will present you methods that allow to try to go further. That is, first, I will present you models that try to go further than the simple PWM model I presented to you. I will also present ways to learn from the DNA sequence itself the regulators that have overrepresented binding sites in the sequence, so that it involves more machine learning aspects. And finally, I will end up, if I have the time, with some perspectives on how this is useful for the interactome and what has been known nowadays as the diseasome. Okay, so let's begin with this first part. So the idea here is that you've got some known data, known binding data for a transcription factor. Okay, so you know where it binds. And given a simple model, you can predict the binding sites that are in the ChIP-seq region. Okay? So this is what you can know initially. And what you want to do is to learn from these binding sites a more accurate model, a better model, with a bit more parameters maybe, and use this model to infer the true binding sites of the transcription factor. And again, why is this important to have the good binding sites? It's because in the end, that's what you want to compare to, for example, mutation events in the genome or things like that. Okay, so the idea of why do we need models of binding is that you've got an observation that consists of maybe 1,000 of sites for a given transcription factor. So these are bound sites, okay? But these 10 base pair sites, you can have 1,000,000 possibility of such sites, okay? So if you wanted to know for each of the 1,000,000 possibilities the probability to be bound for each site, you would need several millions of observations to constrain each number. Here you only have 1,000 observations. So you need to build a model that has only a few parameters compared to these observations that can be constrained from the data, and then that can allow you to predict for any sequence, for any of these 1,000,000 sequences, the probability to be bound. Okay, so the first kind of model I want to show you are maximum entropy models. So a DNA sequence can be viewed as a string of letters that will be A, T, G, or C. Okay? So you can think of spins that have 4 directions, A, T, G, or C, or 4 colors. And the idea is to use some observations that you get from the data and to find the probability distribution that maximize the entropy with the constraints of this observation. So here is the entropy of the distribution. Here is the basic constraint that's the probability sum to 1. And one constraint you can think of, excuse me, I went just a bit too far. One first constraint you can think of is to say that in your data, you know the frequency of a nucleotide at a given position. Okay? So here it's in red, the frequency of the nucleotide, and you ask that your probability distribution satisfy this observation, this average observation. You have a Lagrangian parameter that is a field, okay, hi, and then you can constrain, you can maximize the Lagrangian, and you find what I've shown you before, the PWM model with the different fields that are shown here. Okay, and these are only a few parameters, so it's okay to be constrained. So the second step one can think of, oh god, I'm pressing maybe too much. Okay, so the second step one can think of is to add another observation, which is the denucleotide frequencies. So the frequencies at which you observe pairs of nucleotides. And when you ask that this constraint, this observed constraint is satisfied by your probability distribution, you end up with Lagrangian parameters that are interaction energies between nucleotides. So this is in physics called the Potts model that is widely used in spin glass physics. Here we have 4 colors, and we have interactions that can be between any positions in the sequence. And you can have a lot of interaction parameters, so you need to take care not to add all of them, because then you would overfit. So I will show you, we use some criteria to determine the good number of parameters to add. A simple example to show you what it brings compared to a simple PWM model. So if you say you observe, for example, 2 times AA and 2 times TT, then the observed frequency would be these frequencies. The PWM model, you would count 1/2 80 and 1/2 80, so you would have 1/4 of all possibilities. It would not fit at all with what is observed. But with the pairwise model, you fit what has been observed. So as long as there are only a second-order correlation, you capture them with the pairwise model. And just before I go to the results, let me show you how we try to capture higher-order correlations. So if you want to capture, for example, third-order correlations in your system, you would have to add a lot of parameters. So we thought of another way to do that, which was to say that the protein maybe can bind in several modes, the DNA sequence, and in each of the modes, it would bind with a certain specificity. So, for example, you could say that in your data, there are similar sites, so sites that are similar together, and so we group them together. And then for each of these clusters, there is a PWM that is associated. So the protein, for example, would be either in the red mode, either in the blue mode, and would bind sites in each of the modes with a certain specificity. And here, implicitly, you introduce third-order correlations. So that's a way to simply add correlations, not explicitly, but implicitly, without having too many parameters. Still, you need to have a not too big amount of clusters, so for example, 4 or 5 clusters usually. Okay, so these are models that we wish to constrain with the data, hoping that they would fit better than the PWM model. And so basically the way we fit them is that we have some initial PWM that we use to scan the sequence. We find the sites in red, and we use these sites to constrain either the pairwise model or the cluster model. And in each case, we add one interaction at a time, or here one cluster at a time, until we minimize the Bayesian information criterion. which is basically a maximum likelihood estimator with a penalty that is proportional to the number of parameters, and that allow you to avoid overfitting. And then we take the best model, we scan again the sequences and so on until we converge to some stable set of sites. Okay, so here is a representation of the kind of results that you get, where you compare the probability given by your model for the sites that are observed compared to their observed probability. And what you want is to concentrate over the diagonal. And so in blue, you've got the PWM, a bit scattered. In green, you've got the mixture model that centers more. And in red, you've got the pairwise model. This is for one example, which is the twist transcription factor. And because this is logarithmic scale, you want to look at the upper right corner. And when doing so, here are the most overrepresented sites in your data. So in gray is a decreasing frequency as observed in the data. Now in blue, what you get is a modeled probability by the PWM, by the simple model. So you see that it fails at capturing the observed frequencies in gray. But now when you add correlation, either in the mixture model, either by the pairwise model, you are able to capture the nonlinear decrease observed in the frequency. And actually this is important because overall you end up with about 20% of your predictions that are different between your new model and your simple PWM model. So now we wanted to compare a bit, study the model, what we had introduced, what kind of correlation, and how they would differ between mixture and pairwise. So one thing that we did first was to try to represent them and try to understand what we had added compared to the PWM model. So here would be the simple independent model. And then what we did is that we tried the mixture model. So we have 5 clusters, and for each cluster, we build a PWM. So here in each PWM, you see that there are, for example, different nucleotides in the middle. Okay? So this is the simple mixture model that we've introduced. And we wanted to compare to the, I don't know why. Okay, and we wanted to compare to the pairwise model. And in the case of the pairwise model, you just don't have just one consensus sequence, which is the bottom of your basin and then higher energy state. You have several metastable states in your energy landscapes. So you cannot just build one PWM, but what you can do is to take the sequences that fall in each basin of attraction to a metastable state and build a PWM with those sequences. So that's what we did. We built the PWM corresponding to all the metastable states in the energy landscape. They are represented here. And what I did is I associated each PWM to the PWM of the mixture model with the minimal Kullback-Leibler divergence. So here is the metastable PWM, and here these are the mixed-up model PWM. And what you can see is that actually, in the pairwise model, you can faithfully associate the PWMs you obtain with a PWM from the mixture model. So in this case, you have a strong interaction in the two nucleotides in the middle, and therefore you capture this strong dinucleotide correlation with your pairwise model, and the mixture model does not capture higher order correlation. So the pairwise model is sufficient to capture correlations. So then what we did is that we wanted to quantify these interactions that were introduced, and we used to quantify it a quantity that was introduced previously in the case of trying to find direct interaction in protein folding, and that is called direct information. It's like mutual information, but with mutual information, you basically just use correlation between two variables. So this correlation can stem from indirect links. Here it's built from the interaction parameters that quantify only the direct contribution. And we normalize this direct information in such a way that it's between 0 and 1. And so here is the interaction matrix. And for example, here you can see that this position interacts a lot with the previous position here. So we end up seeing what we observed just previously, that these two guys are interacting a lot, even if in the PWM you don't see it. You don't see that there is a lot of information here. And couplings are about 10% of the maximum they could be. So you introduce a lot of weak couplings. Still, they are weak, but higher than in the background of the genome. But they allow to fit the data better, which is something that was a kind of message that was already given by Bill Bialek, for example, in the case of neural networks. And what is nice with such a general model with interaction of any length is that you can actually see what is important in your model. And for example, if you look at the strength of the interaction as a function of the distance between nucleotides, you find that in the different transcription factors that were studied, it's mainly nearest neighbor interactions that you need to add. So that allows, this is general enough to show you what is necessary to add. And moreover, you can use that to see where in the binding sites you usually add information. And so here in blue, I show the interaction strength. And I've shown that here it's mainly in the flanking nucleotides of the sequence that there are interaction. And that's mainly where the PWM has low information. So you add information where there was no information, which could explain the improvement in prediction. Oh, excuse me, I think I pressed it twice. So just to give you a sense of this first part, what I've shown you is that it's possible to improve the prediction of transcription factor binding sites in vivo just by introducing some pairwise correlation with this POTS model. And actually, nearest neighbor would be sufficient, so you don't have to add so many parameters. And it's important if you want to associate these binding sites with mutations in the genome to understand which part in your regulatory network are mutated. There are substantial differences between the predictions, so again, this is important for this reason. And now a lot of data has become publicly available. So this is data from the ENCODE project that you may have heard about. So last year, they published a lot of papers in Nature and Science with a lot of data rendered public. For example, they have this kind of data for almost 200 transcription factors now in humans, so you can constrain such model and and gain more generality. And all that is public and available on the web, so you don't have a lot of things to do, basically just download. Okay, so this case was building model from known sites. So now is a different part where this time you don't know the factors that regulate the regulatory region, okay, that bind the regulatory region, but you just know that there are pieces of DNA in the genome that are in the non-coding genome, but when you put these pieces of DNA in front of a reported gene, then they will drive some expression in a specific spatial temporal way. So you know that there is information in these pieces of DNA, and you want to get it, you want to learn it, okay? So basically what you want to have is to find, in those pieces of DNA some overrepresented binding sites for transcription factor, and the approach is called de novo transcription factor finding, because you don't use any a priori on this transcription factor, you just try to find the models that correspond to them just from the DNA sequence. And this kind of work is important just because almost half of the transcription factor don't have any models, so you expect to lack information if you use the databases. Then using what you have learned, you want to predict new region in the genome with the same regulatory potential, so with the same output. So this is really pattern recognition. You want to learn from a few examples something that is general enough to allow you to predict on the genome scale pieces of DNA with the same output. Okay. So we build an algorithm to do that is called Imogene. Well, so you, as I told you, you begin from DNA sequences that have expression localized with some specific pattern of expression. Then, because usually you don't have a lot of data, it's very hard to obtain. Maybe when you collaborate with biologists, you have maybe 10 or 15 such sequences with knowledge of the regulation. So you want to increase the size of the data set. And to do that, we go in many related species to find the corresponding orthologous sequences. Okay, so that's allowed to increase the size of the data set by a factor of 10. And then we have a motif builder, which I can explain more during the question if you want, where basically we scan the training set. So we scan the training set in the reference species. From each possible starting point, we build an approximative PWM, this shown here. So we build an approximative one. We scan all the sequences to detect putative binding sites. And then we use all these binding sites to constrain the model in a refined way, and so on until we reach convergence. And compared to previously, when you learn the model, it's more difficult because here the species are related by a phylogenetic tree. So for example, very nearby species, the binding sites will be the same because it didn't have time to mutate. While for very distant species, it will have time to mutate, so these are independent observations. So we use an evolutionary model to take into account, to weigh the different observation as being independent or correlated. Okay, so this way we generate a set of PWMs that correspond to overrepresented motifs in the data set. We rank them by the overrepresentation in the training set. So we have, for example, 5 very important PWM corresponding to important factors regulating those sequences. And then we use this PWM to scan the genome and predict sequences where you've got a clustering for binding sites for this PWM that are conserved among the related species. Okay? You predict CRM, and then you want to test for their activity and want to see if your prediction is right. And so I will present you first results just in silico from cross-validation showing the sensitivity, the ability of this program to find bona fide CRMs. And after that, I will show you experiments that have been done to actually validate that the motifs found are functional, to show you that it's just not random. Okay, so the first experiment, the cross-validation, is taken from a benchmark from those guys in the SINA lab. Their idea was to provide a benchmark consisting of sequences that are specific to either to neural tube or to the limb development in the mouse. And basically, you have, say, 20 cis-regulatory modules in each case. You take one out, you learn on the other CRMs, and you try to predict the left-out CRM in 10 kb background. So if it was just random, you've got a 1 kb piece of DNA in 10 kb background, you would have prediction rate of 15%. And now you want to see if your tools allow to go higher than this prediction rate. And here I will compare the methods that we have developed to methods that are called motif blind, because they do not use PWMs, but they use stuff that are more like signatures of CRMs that are based, for example, on Markov chains of order 6 or things like that capture some patterns in the sequence, but you cannot associate them to a motif. And I wanted to compare to these methods because they are known to work better than methods that relies on motif from the database. So I wanted to see how much the de novo methods would bring as information. So here you can see the results for neural tube and limb. And shown is the sensitivity of the different methods. So above the line means significant. And here is Imogene, and here are the different motif-blind methods. So we are happy to see that at least it performs as well as those methods. But the thing is that compared to those methods, you also have other information to bring to the biologists that you find motif that actually corresponds to some bona fide motifs. So for example, in the neural tube case, here are the third and fifth motif generated, and you can see that they correspond to known motifs in the database that are known to be involved in neurogenesis. So it's not absurd, but beyond this motif, you also find de novo motif. And now I want to show you some tests of this de novo motif to show you that they can be interesting as well. And these tests happen in Drosophila. So we have collaborated with a team in Toulouse that work on the development of hairs in Drosophila. These hairs are called trichomes. And their pattern and their differentiation rely on a gene that is the master gene of the differentiation that is called shaven baby. Because basically, if you mutate it, the baby is shaven, I guess. And there was already some known PWM for this gene, okay? A known motif for the transcription factor. So the team, before we were involved, used this known motif to scan the genome, predict regulatory sequences, and they actually tested the prediction and found that only 1/3 of them was working, was having the good pattern of expression. So we wanted to see if Imogene could succeed in better classifying, better finding positive sequences from negative. So we generated motifs from the positive sequences, and we kept the motifs that were best at distinguishing positive from negative sequences. The idea is to find a couple of motifs that would really be overrepresented here and not here, because you see that previously the OVO Q6 motif was found everywhere. Okay, so we found two motifs. The first one here is a refined version of this previously known motif. And the other one we call the blue motif, because I used the blue color to find it. And it's new, and we could not find anything in the database corresponding to it. So first we tested the refinements involved, for example, here by the presence of this information content on the A in the beginning of the site. And by point mutation, this single nucleotide was shown to be important for the correct expression of a reporter of the expression of a regulatory region containing a binding site for this transcription factor. And the de novo motif was mutated in 3 different regulatory regions and abolished expression in all cases, which was a good validation of the importance of de novo motif. And then, as I said previously, you can use this motif to scan the genome, find some regulatory regions that are similar to the training set. We combine that to binding data for shaven baby and with expression data. And this case, we could attain 80% validation rate. And when I say validation, I mean that the sequence reproduces the pattern of expression of the trichomes, and that in shaven baby mutant, you don't have expression anymore. And one thing that we saw is that there is no unique grammar of the inputs that lead to a same output. So there are different ways to do the same thing in these regulatory regions. Okay, so to finish with Imogene, we rendered everything public on the website, so biologists can input their coordinates, find motif and scan sequences and predict regions in the genome. Okay, so to get over with that, here what I've shown you is that we have an algorithm that allows to find new motifs from DNA sequences and that predicts as well as methods that are better than methods based on known motifs only. So hopefully we bring new information. The de novo motif seems functional. You don't know what transcription factor it is associated to, but this could be done with some biological experiments that can be achieved that are called one hybrid experiments. I can describe it to you if you want later on. Okay, it's on the web. So how this basically, if we zoom out a bit, where are we now, considering like the regulatory network and potentially the disease network? So what I've shown you is that we can find transcription factor binding sites, but for now I didn't talk about linking to diseases. And as I told you in the beginning, most of the mutations associated with disease are in non-coding sequences, so are outside of the protein coding regions. So, and I said earlier, half of them actually are in transcription factor binding sites. Second thing I did not show yet is how to link what you found, so the transcription factors that regulate a site, how to link with a target gene. Usually what we do is that we link to the nearest target gene, but actually it's known that it's not always the case. So how to know that? And what I want to show you as a perspective is how now, thanks to the ENCODE public data, it becomes possible to use some recent experiments to actually get this information and combine with the previous information to really get to the regulatory and disease network. And this kind of experiment is called a DNase 1 experiment. It has been an experiment known for a long time in biology, but now it's only recently that it's been developed on a genome-wide scale. The idea is simple, is that there is an enzyme, DNase 1, that cuts DNA in open region. So as soon as you have a region that is not bound by a nucleosome or by a transcription factor, it cuts it. So you can get those cut sequences, you can sequence them, you can align to the genome, and what you see is that here are DNAs that has cut the genome in this region. So this is a promoter of a gene where actually NRF1 protein was bound. Okay, and if you zoom in this region that was cut, you can zoom in until you find a small region, approximately 15, 20 base pair, where it is not cut. And that's where the protein is bound. And when you further look at the sequence, you can actually find, with nucleotide resolution, the binding sites that is associated to this binding. And so what is important is that in this experiment, you just know something is bound, and then you've got to look in your motif, in your database, anything, what is the best putative motif that binds to it. Therefore, it's important to have good models. And actually, using this data alone, this available data alone, you can know if your transcription factor can be mechanistically caused, linked to a disease. So here is an example of a variant, a mutation, okay, that basically this is a mutation where you will have a C on one allele instead of the T. And here are the DNase reads that come from the chromosome where there is a C. And you can see that the region is being cut. Indeed, when you compare to the motif, you see that the nucleotides here are not the same. C is not T, so basically you have difficulties to bind. But here what you see is that if on the chromosome where you've got the good allele, you've got exactly the good nucleotide, so you are bound by the factor. So here what you see is that actually just having the SNPs, so having these mutations, having this DNA's signal that is available, and having the model for the transcription factors, you can detect what effects a mutation can have, which transcription factor is involved. And the second thing, now, so you know what transcription factor binds, and potentially what disease may be linked. And now you want to associate with the target gene. And here is an example where actually you can associate the binding event to a target gene. And the idea is that the DNA's signal, you have that in more than 100 cell types, in hundreds of cell types. And if you look in different cell types, here you see that you will have a cell-specific binding event that is not reproduced in other tissues. There are ubiquitous signals here. And here, again, you've got a signal that is cell-specific here, but not here. And so basically, you can compute a correlation between DNA's traces that are in non-coding region and in promoter region. So here it corresponds to the polymerase that is bound, and here to a regulatory region. And therefore, you can actually associate the regulatory region with the target gene. And actually, not most of the time, but at least more than 1/3 of the time, it's not the nearest gene, but it's jumping over a few genes. Okay? So here, okay, just to give you some food for thought, how this data can be linked to bigger networks. First, I've shown you that we have better models to predict the DNA regulatory sequences, and they can be used to actually combine with the DNA's footprint data to find the, so we have the upstream regulator, we have the downstream regulator, which was explained just before. So this allows to build a regulatory interactome, not only proximal, but on the genome-wide scale. This can be used as well to see which transcription factor in the regulatory interactome are affected by mutations. And then you can build what is called a diseasome, where you will have related, so it's a network of disease, where disease will be related if they share, if the mutation associated to the disease actually fall in binding sites for similar transcription factors. You have all that alone with just with DNA sequence and models. You have insights on the causality of SNP just by seeing if the footprint is affected by the allele in the two alleles. And then you can also think of crossing this data with some known regulatory region and learn de novo motif and see if these de novo motif actually fall in some SNPs or so some disease that for now have no transcription factor associated to. Okay, so this was more to take a step back and give some perspective. But for the rest of the work, so these were people I'd like to thank. So physicists from the LPS, Vincent Hakim and Thierry Morat. So with Thierry, we built this inverse problem, this POTS model to describe the binding of the transcription factor on DNA. Hervé Rouault, also a physicist, and François Schweizgut, a biologist, with whom we collaborated to build this de novo motif research. And this team in Toulouse, with whom I worked to test and validate some of the things that were found. And thank you to you, and I hope it was not too much biology for today.
Thanks, Marc. Questions?
When you build your POTS model, you use first the one-body interaction to compare with the single point distribution, you obtain the H's, the H set, then you test the two-body correlations, and then why do you don't use the third body, the fourth body?
So the problem
comes to the
absolutely, it's a mixture model. So the thing is that already with the pairwise model, you have 1,000 parameters, and you've got 1,000 observations. So already here, you need to take care of the overfitting by not including everything. So we feared by going to third order correlation, where you've got basically QL to the third numbers. So basically, you begin to have some 10 to the 4, 10 to the 5. It cannot just be constrained by the data systematically. So then, on a case-by-case basis, one could think of introducing one or two third-order correlation. But so we did not do that because we thought that these kind of things could be captured by the mixture model. And because we did not see significant differences between the mixture model and the pairwise model, we decided to stay at this point. But would the mixture model have given some insights on higher order correlations, then we would have taken a step in that direction. Yes.
More questions or comments? Marc, you said that at the beginning that biology was an excuse here. And that was one of your favorite sentences. My question is, I mean, you've developed here a number of methods and tools. In which sense do you think that these methods can be useful in other fields?
Well, basically, the tools...
For which of them could be used?
Well, clearly, inverse problem is basically... both tools are very general in the sense so that the inverse problem is just trying to find interactions in a network. Okay, so here it's a small network of 12 nucleotides, and you want to find the interaction between nucleotides. So this is a very general problem, and it can be done on larger networks with some approximation, like mean field approximation or things like that. But so this kind of tool is very general. It's just applied on DNA sequence bound by the transcription factors that will apply a field and apply interactions. In the second case, it's basically inference problem, where you, this is trying to get from the data in a Bayesian way some patterns. So here it's just you infer a simple model, the PWM model, in a very crude way with some priors and from one observation, which is your starting site. And then you use this prior, a bit refined prior, this approximative prior, to scan the sequence and refine your prior in a Bayesian way by the observation to build a posterior function that then is refined and so on until, well, finally we take the maximum of the posterior to have the model. But it's also model fitting from data, and it's basically most general. It's just that the model and the data is peculiar to this biology realm. That somehow it's not. I mean, this is pattern recognition really in the end. It's model fitting to data, constraining parameters, taking care you don't have too many parameters. And the thing that is nice with biology, in some sense, is that then you do this pattern recognition, but you can actually not only predict, but in the end make mutations or tests that this pattern recognition works well. And well, so that's.
In the first case, when you use the post model, you feed the post model with some data, you obtain the parameter.
Yeah.
So what is in the post model that is not, for example, in a NOVO network that makes more suitable to infer that? You can have a NOVO network, you can train it with some data and see if whatever it predicts. So I guess that the post model works better than a NOVO network, so there should be something in there.
I don't know if it works better, because, so for example, even the PWM, when I compare that compared to other methods that could use, for example, even Markov chains of order 6 or other types of supervised or non-supervised learning. But basically here the idea is that it has a physical meaning. what you introduce in the end, you expect that this is an interaction energy that is due to some structure effect, and then you can call your best crystallographic friend and tell him, hey, do you have data for this type? I wonder if the protein has something. So because in the end, we want to link that a bit to the physical realm, having interaction energies is a nice thing. As a physicist, it's very supervised. And so you know that what you introduced, you have some expectation that they have some meaning in the real life.
But you don't introduce the parameters of these interacting energies. You just introduce so you guess of this interacting energy. You don't use, for example, what is the interacting energy between that molecule or that molecule or that other one, that base on that other base. You don't use that. You infer things from the data. You could say, well, if I have a guanine here and a thymine here, the interaction could be, I don't know how many calories or whatever, or trombones or whatever you want to, or you measure the energy, there. You don't use that. You don't use the binding of the energy. Because you don't know what is the structure of the two parts of the molecule that they do interact. There are many bases that are there, and the interaction is not just a sum of interacting energy.
That's true. So that's true. And so the way to get that would be to compare with crystallographic binding. Well, it would be in vitro, but crystallographic binding with basically mutations, gene nucleotide mutation, and seeing the effect and seeing the variation in structure and trying to understand mechanistically. That says that would support mechanistical investigation. That's what I want to say. I don't use it, but that would support it, and that could be used for it.
But still, what it works better than crystal than just assuming nothing is the fact that you assume interaction of energy somehow, in some way, with some parts, I guess.
Yeah, no, but maybe, I mean, I could try a different, really like neural network.
Let me put the question another way. If you take the parameters that you have inferred for this parameters for the Potts model, since some of these parameters are interaction energy,
so you can fit that. So here you don't have the scale of the energy, but then you can use mutational experiments and gel shift or things like that. That would you need experimental data, and then you can see if your model fits the experimental data with the inclusion of your predicted energies. So, I mean, that's an experiment that is doable.
Another question about this Imogene algorithm. Again, you use Bayesian data to infer or Bayesian statistics to infer what is the correct parameter of the model. So I guess, and you compare with other ways of doing the same, and it seems that this is better than other ways to infer what are called these transcription factors and all that. So I guess that the others also use Bayesian data because Bayesian statistics is the only way to infer something from data is always using Bayesian. So what is in Imogene that is not in the other ones that allows it to provide better results?
So here, so the other one first, the kind of things that they used are okay, not really Bayesian. So there is this non-motif stuff, which is another thing. Okay, so for example, they use collections of words, overrepresented words from the data. Okay, so that would constitute somehow a word budget with which you want to compare a given sequence to see if it corresponds to that word budget. But the thing is that with this kind of models, again, because it is abstract, you don't actually classify these words in different PWMs, in different models, but you have just a bunch of words. Here, the difference is that you end up with some cluster of words that correspond to transcription factors. And then you can associate that to the database or say it's de novo. The idea is mainly, first, there is the clustering. So there is the fact that we know that we associate to something. And second, it's these models that they use, they do not rely on evolution. So they don't use these evolutionary models and stuff to build the PWM. So somehow we use a bit more data. This refining process, introducing an evolutionary model to build and to weigh the difference observation is not something that is used. Usually, for example, people take everything as independent observation, and this basically just overfits, because sometimes you have strong conservation because just you've got the rat and the mouse and they are the same, and just you've got two times the same size. So this kind of thing is not appreciated in these other algorithms. But if you want me to show the exact evolutionary model or how we do the computation, I'd be glad to show you if you want. That's the main point.
Any other comment or questions? Well, if not, thanks very much, Marc.